Welcome to Curiosity, the podcast where science meets wonder. Join us as we explore the fascinating, the innovative, and the unexpected, from the secrets of the cosmos to the marvels of everyday life. Each episode takes you on a journey to uncover the knowledge and stories that fuel our endless quest for understanding. Let your curiosity guide you.
The Microbiome Revolution: How Gut Bacteria Shape Health and Disease. The human body is a remarkable symbiotic ecosystem, harboring trillions of microorganisms that collectively form the microbiome. These microscopic residents, predominantly bacteria, reside in various niches within the body, with the gut emerging as a particularly dense and diverse habitat. In recent decades, the field of microbiome research has revolutionized our understanding of human health, shedding light on the profound interplay between gut bacteria and physiological processes. Far from being mere passengers in the digestive system, these microorganisms actively contribute to digestion, immune modulation, and even the regulation of behavior. This evolving field has revealed the microbiome as a dynamic entity, influenced by diet, genetics, environment, and lifestyle, capable of both bolstering and undermining health depending on its composition and interactions with the host. The gut microbiome's influence extends beyond the confines of the gastrointestinal tract. As scientists delve deeper into its intricacies, a startling picture emerges of its role as a mediator of systemic health. One of the most striking discoveries is the gut-brain axis, a bidirectional communication network linking the central nervous system with the enteric nervous system and gut microbiota. This connection highlights how gut bacteria can influence neurological and behavioral outcomes, with evidence suggesting their involvement in anxiety, depression, and even neurodegenerative diseases like Parkinson's and Alzheimer's. These findings challenge traditional views of mental health and emphasize the microbiome's role in shaping not just physical but psychological well-being. Equally compelling is the microbiome's impact on the immune system. A balanced and diverse gut microbiota is essential for the proper development and functioning of immune defenses, acting as a training ground for immune cells. It teaches the body to distinguish between harmless substances and harmful pathogens, thereby preventing autoimmune responses and chronic inflammation. Conversely, dysbiosis, a state of microbial imbalance, has been linked to a host of conditions, from inflammatory bowel disease to allergies and metabolic disorders like obesity and type 2 diabetes. This intricate relationship underscores the importance of microbial health, as disruptions in this balance can have cascading effects throughout the body. Microbiome research has also unveiled its critical role in metabolic processes. Gut bacteria are involved in breaking down complex carbohydrates, producing short-chain fatty acids, and synthesizing essential vitamins like B12 and K. They influence energy harvesting from food and impact fat storage, pointing to their significant role in conditions like obesity. Moreover, emerging studies suggest that certain bacterial species can modulate blood sugar levels and lipid profiles, offering potential avenues for the prevention and treatment of metabolic syndromes. As such, the microbiome is increasingly recognized as a key player in nutrition and metabolism, its composition acting as both a marker and a determinant of overall health. The implications of these findings are transformative, reshaping approaches to medicine and public health. The prospect of microbiome-based therapies is particularly exciting, with probiotics, prebiotics, and fecal microbiota transplants, FMT, garnering attention as potential interventions. These strategies aim to restore microbial balance, offering new hope for patients with conditions that were once thought to be unrelated to gut health. The potential to manipulate the microbiome to prevent or treat diseases has catalyzed a wave of innovative research, seeking to decode the complex interactions within this microbial ecosystem. Scientists are not only identifying key microbial players but also exploring the specific metabolites they produce, which act as signaling molecules influencing host physiology. As microbiome research continues to evolve, its scope expands, raising profound questions about the nature of health and disease. What determines a healthy microbiome? How does microbial diversity correlate with resilience against illness? Can we precisely engineer microbiomes to enhance human health and longevity? These inquiries lie at the heart of a burgeoning scientific frontier, one that promises to redefine our understanding of biology and medicine. The gut microbiome, once overlooked, is now at the center of a revolution, its influence touching nearly every aspect of human life. The deeper scientists delve into microbiome research, the more intricate and interconnected its influence on health and disease becomes. One of the most captivating areas of investigation lies in its role in early development. From birth, the microbiome begins to take shape, profoundly influenced by the mode of delivery, maternal microbiota, and infant feeding practices. Babies born vaginally acquire a microbiota dominated by beneficial bacteria like lactobacillus, while those delivered via cesarean section often exhibit a different microbial profile that may predispose them to allergies, asthma, and metabolic disorders later in life. Breastfeeding further enriches the gut microbiome, as human milk oligosaccharides, HMOs, act as prebiotics, selectively feeding beneficial bacteria such as Bifidobacterium. These early interactions between host and microbiota set the stage for long-term health, underscoring the importance of microbial exposure during critical windows of development. As life progresses, the microbiome continues to evolve, shaped by dietary habits, environmental factors, and lifestyle choices. Diet emerges as one of the most powerful modulators of microbial diversity and composition. Fiber-rich diets, for example, promote the growth of bacteria that produce short-chain fatty acids, compounds that have been shown to reduce inflammation and support gut barrier integrity. Conversely, diets high in processed foods, sugar, and saturated fats can foster dysbiosis, contributing to chronic inflammation and metabolic disturbances. This dynamic relationship highlights the importance of dietary patterns in shaping the microbiome and, by extension, overall health. It also explains why shifts toward westernized diets have been implicated in the rising prevalence of obesity, diabetes, and cardiovascular diseases worldwide. The influence of the microbiome extends beyond digestion and immunity, playing a pivotal role in chronic diseases that were once considered unrelated to gut health. In cancer research, for example, certain bacterial strains have been found to modulate the efficacy of chemotherapy and immunotherapy, either enhancing or hindering their effects. Fusobacterium nucleatum, a bacterium commonly associated with colorectal cancer, has been shown to accelerate tumor growth and resistance to treatment. On the other hand, some gut bacteria appear to enhance the body's immune response to tumors, suggesting that manipulating the microbiome could improve therapeutic outcomes. These findings open up new avenues for personalized medicine, where the microbiome becomes a target for optimizing cancer treatments. Autoimmune diseases, such as rheumatoid arthritis and multiple sclerosis, also exhibit strong links to the microbiome. Emerging evidence suggests that dysbiosis can trigger aberrant immune responses, leading the body to attack its own tissues. For instance, specific bacterial species have been implicated in the breakdown of immune tolerance, a critical factor in the onset of autoimmune disorders. This connection offers the tantalizing possibility of using microbiome-based therapies to re-establish immune balance, potentially preventing or mitigating the progression of such conditions. Researchers are increasingly exploring how probiotics, dietary interventions, and even microbiome transplants could serve as non-invasive treatments for autoimmune diseases, offering hope to millions of patients. The gut-brain axis remains a particularly fascinating domain, as scientists uncover how microbial metabolites, such as neurotransmitter precursors, influence brain function and mental health. Serotonin, often called the happiness hormone, is primarily produced in the gut, and its synthesis is closely tied to the microbiota. Studies have shown that changes in gut bacteria can alter stress responses, anxiety levels, and even cognitive function. Animal models have demonstrated that transferring the microbiota of an anxious mouse to a germ-free mouse can induce anxiety-like behaviors, providing compelling evidence of the microbiome's role in shaping behavior. Human studies are now beginning to reveal similar patterns, with alterations in gut bacteria linked to depression, autism spectrum disorders, and schizophrenia. These discoveries are reshaping our understanding of mental health, suggesting that therapies targeting the microbiome could complement traditional psychiatric treatments. The interplay between the microbiome and infectious diseases further underscores its far-reaching impact. The gut microbiota acts as a critical line of defense against pathogens, competing for nutrients and producing antimicrobial compounds that inhibit their growth. However, disruptions caused by antibiotics or illness can weaken this defense, allowing opportunistic infections like Clostridioides difficile to thrive. Restoring microbial balance through fecal microbiota transplantation has proven remarkably effective in treating recurrent C. difficile infections, highlighting the therapeutic potential of microbiome modulation. Beyond the gut, researchers are investigating how microbiota in other parts of the body, such as the skin and respiratory tract, influence susceptibility to infections, offering a more holistic view of microbial host interactions. As the connections between the microbiome and health become increasingly clear, ethical and practical challenges emerge in translating these discoveries into clinical practice. The complexity of the microbiome, with its immense diversity and variability among individuals, presents significant obstacles to developing standardized treatments. Moreover, the prospect of microbiome engineering raises ethical questions about the long-term consequences of altering such a delicate ecosystem. Despite these challenges, the field continues to advance rapidly, driven by cutting-edge technologies like metagenomics, metabolomics, and single-cell sequencing. These tools enable researchers to analyze the microbiome with unprecedented precision, unraveling its vast complexity and identifying specific pathways through which it influences human health. The final frontier in microbiome research lies in its potential to revolutionize precision medicine and redefine how we approach health and disease. The microbiome's inherent uniqueness, shaped by individual genetics, environment, and lifestyle, demands highly personalized strategies. Unlike traditional medicine, which often takes a one-size-fits-all approach, microbiome-focused therapies have the potential to account for this variability, tailoring interventions to the specific needs of an individual's microbial composition. For instance, probiotics that may benefit one person could prove ineffective or even harmful to another, depending on their existing microbiota. As such, the future of microbiome-based treatments relies on the development of diagnostic tools that can accurately profile a person's microbiome and predict how it will respond to dietary changes, medications, or therapeutic interventions. One of the most promising avenues for therapeutic innovation is the use of microbiota-derived metabolites. These small molecules, produced by gut bacteria, act as chemical messengers that influence a wide range of biological processes, from immune function to neurotransmitter synthesis. Researchers are now exploring ways to harness these metabolites as drugs, bypassing the complexity of manipulating entire microbial communities. For example, short-chain fatty acids like butyrate, which have anti-inflammatory properties, are being studied for their potential to treat inflammatory bowel disease and other chronic conditions. Similarly, microbial-derived bile acid derivatives are being investigated for their role in regulating metabolism and protecting against liver diseases. These breakthroughs highlight a shift in focus from merely cataloging microbial species to understanding their functional contributions to health and disease. Another transformative application of microbiome research is the development of engineered probiotics, genetically modified microorganisms designed to perform specific tasks within the gut. Unlike traditional probiotics, which often have limited and inconsistent effects, these engineered strains can be programmed to produce therapeutic compounds, detect disease markers, or modulate immune responses. Early trials have shown promise in using engineered probiotics to treat conditions like phenylketonuria, a rare genetic disorder, by breaking down toxic metabolites in the gut. This cutting-edge approach blurs the line between biology and technology, showcasing the potential of synthetic biology to reprogram the microbiome for therapeutic purposes. Beyond treatment, the microbiome also offers unparalleled opportunities for disease prevention. Longitudinal studies have shown that changes in the gut microbiome can precede the onset of diseases such as type 2 diabetes, colorectal cancer, and even neurodegenerative disorders. These findings suggest that the microbiome could serve as an early warning system, enabling the development of non-invasive diagnostic tests that detect disease at its earliest stages. For instance, researchers are exploring the use of microbial biomarkers in stool samples to identify individuals at high risk for colorectal cancer, potentially reducing the need for invasive procedures like colonoscopies. The ability to predict and prevent disease through microbiome monitoring represents a paradigm shift in healthcare, moving from reactive treatments to proactive care. As the field continues to advance, it is also uncovering profound insights into the evolutionary and ecological dimensions of the human microbiome. The coevolution of humans and their microbial companions over millennia has shaped not only our physiology but also our adaptability to different environments and diets. For example, populations with diets rich in seaweed, such as those in coastal Japan, harbor gut bacteria capable of breaking down complex polysaccharides found in marine algae, a capability absent in most other populations. These findings underscore the deep interdependence between humans and their microbiota, highlighting how changes in diet, environment, and lifestyle can disrupt this delicate balance. The rise of urbanization and the widespread use of antibiotics have significantly altered the human microbiome, potentially contributing to the global increase in chronic diseases. Understanding these shifts is critical for developing strategies to restore and maintain microbial health in an increasingly industrialized world. However, the promise of microbiome research is not without its challenges. The complexity of microbial ecosystems, coupled with their dynamic nature, makes it difficult to establish clear cause and effect relationships. Much of the current knowledge is derived from correlative studies, and translating these findings into actionable interventions requires rigorous clinical trials. Additionally, the ethical implications of microbiome manipulation, particularly through technologies like gene editing and fecal microbiota transplantation, raise important questions about safety, consent, and the potential for unintended consequences. As researchers strive to unlock the microbiome's full potential, they must navigate these challenges with caution, ensuring that scientific progress is guided by ethical principles and a commitment to improving human health. The future of microbiome research holds extraordinary promise, offering a glimpse into a world where health and disease are understood through the lens of microbial ecology. By bridging the gap between microbiology, immunology, neuroscience, and medicine, this field has the potential to transform our understanding of what it means to be human. The microbiome is not just a collection of microorganisms, it is a dynamic and integral part of our biology, shaping our health, behavior, and resilience in ways that we are only beginning to comprehend. As science continues to unravel its mysteries, the microbiome stands poised to redefine the boundaries of medicine, offering new hope for a healthier and more personalized future.